for the opportunity to be here talking about my data today. It's very wonderful. And today, as you might expect, I will talk about my recent paper on PNAS that will basically describe how ILC3s are very important for controlling neutrophil response during entering infection. So the project is very straightforward. I think it will be very easy for everybody to understand. We did a lot of things, but I think the things that we did is self-explain the paper, so it's very really straightforward. And I hope everybody can get the message. And of course, I'm always open for questions at the end. And so the whole project basically started knowing the biology of ILCs. So what is ILCs? We know that ILCs are lymphocytes because they have common progenitor with T cells and B cells, but they don't have TCRs. So because of they lack TCRs, they don't respond to antigen. So their activation basically rely on cytokines produced by the signals on the tissue. And they mirror the CD4 T8 cells. So for example, we have ILC1s that mirror T8-1, producing interferon gamma. You have ILC2s that mirror Th2, producing IL4, 5, 13. And in special, we have ILC3s that mainly produce IL-17 and IL-22 that mirrors Th17. Specifically about this ILC3 or group 3 innate lymphoid cells, they're mainly found in the gut mucosas. So you can have a lot of ILC3s in the small intestine and also in the colon. And they sense very well the signals from like the early inflammation or early danger signal as cytokines, as IL-23 and IL-1 beta. In response to the signals, they produce mirroring the type Th17 cytokines. They produce IL-22 mainly and IL-17 and another cytokines. Overall, these cytokines are very important for priming the tissue, mucosal tissue function. So we can see antimicrobial peptide expression, tight junction protein expression, mucus secretion, epithelial cell proliferation, and they also have a little bit of role on immune cells. For example, IL-17 is known to induce neutrophil migration to the tissue and then cause this very acute innate inflammation. And they produce these cytokines while the dendritic cells or the antigen-presenting cells are priming the T8 cells to be activated and produce those cytokines. So it's a very fast way to release these adaptive cytokines of the tissue in a TCR-independent manner. And because of this very fast protection as played by these ILC3s, they are known for being involved in many enteric infections. So there are many diseases. I put some here, but of course, if you go to literature, you can find many others pathogens that cause activation of ILC3s in the context of infection, and they play a very important role for containing these pathogens. I want to address attention to one of these bacteria that is basically Clostridioides difficile. That is a bacteria that causes infection in patients under antibiotic therapy. So it's an antibiotic-associated disease. And it's causing many infections in healthcare settings because people go there for treating another disease, like I don't know, like pneumonia, another infection, and they have antibiotics for the treatment of this infection, and then they get their acquire C. diff, and then they start having this antibiotic-associated C. diff infection. It's a very strong disease, affects millions of people worldwide, and the death rate is very high. So a lot of people die yearly because of this infection. And it's basically caused because C. diff is one of these superbugs that are resistant to many antibiotics. So we are facing now many strains of bacteria that are resistant to antibiotics. And they don't die very easily. So we have this very strong infection that are persistent and in many cases becomes inflammatory systemic inflammation and the patient die. So C. diff is one of these super bacteria. And because it's resistant to antibiotic, when you have antibiotic treatment, you disrupt the normal microbiota. And the normal microbiota is known to avoid C. diff overcolonization in the mucosa. And so without the microbiota, C. diff overgrow. And this overgrow is associated with the release of two toxins, A and B, that mainly kill the epithelial cells, this interruption the epithelial layer. And because of that, it's obvious we have inflammation, we have bacterial translocation, and eventually systemic inflammation that will lead to death, basically. In a mouse model, this infection is very similar to human. You have an antibiotic treatment, you have a disruption of the microbiota, and then you have the very acute disease. But unlike human, the infection in mouse only happens for 1 week, and then the mouse recovers, and you don't have the disease anymore. Eventually, 1/4 of these mice can recover the infection because it's a recurrent relapse infection, and they can get sick again. But the main moment of the infection is the acute early phase of the infection. We know that it's very similar to human. So we have the bacterial colonization, secretion of cytokines, and then we have the epithelial disruption, a very strong bacterial translocation, inflammation mainly associated by neutrophil infiltration, and then in 5-6 days, you have the healing process. So basically the cells start cleaning the tissue, CGF the number goes down, and then you have less cytokines, and then the more homeostatic cells start cleaning and healing the tissue in a resolution phase. We published a few years ago with my previous lab in Brazil in collaboration with Dr. Colonna here in US. And this work basically showed that the microbiota is important for avoiding C. diff colonization and infection because they produce short-chain fatty acid, and this molecule can coordinate the innate immunity in the lamina propria. One of the main finds we found was that ILC3s was affected by this short-chain fatty acid, this metabolite, and they produce high level of IL-22. And these cytokines are very important for inducing the epithelial healing. So the epithelial cell proliferate, they produce antimicrobial peptides, and then they induce this recovery from the C. diff infection. And then when we block IL-22 with antibody or using a knockout mice, we don't see this recovery anymore. So the mice are still sick at the end of the infection. And this was kind of confirmed by another group by Eric Palmer from Chicago. They showed that IL-6s are very important for the late phase of the infection, where they produce a lot of IL-22 that is very important for the epithelial healing. But they also show in their paper that ILC1s are very important for the early stage, that day 1 or 2 post-infection, producing mainly interferon gamma. And then we decided to investigate if ILC2 can also be important for the early stage, because we know that they are there. We have a lot of increased amount of ILC3s in the early phase. So we checked the lamina propria after 2 days of the infection, as you can see here by gating all the ILCs. And you can see the ILC1 was higher, as reported by Eric Palmer group, but ILC3 was also higher after 2 days of infection. So they are there, but they are only suggested to be important in the late phase when they produce IL-22. But since they are there and they strongly correlate with neutrophils, that they are the main cells that infiltrate in the tissue, we start wondering, are these cells important for the late phase? So as I show here, so they are there and they correlate with neutrophils. So are they important? For investigating this, of course, as scientists, we all know that we need a model with depletion of the pathway or the cell. So since we want to investigate the early impact of ILC3s and the C. diff, we need to deplete the cells somehow. We can use antibodies, we can use like genetic modified mice, but we're still looking for a model. So then we decided to investigate the literature. Our lab reported in 2019, if not wrong, or like a few years ago, with another group from Chicago as well, that AHR is a transcriptor factor, very important for the development of ILC3s. And when you don't have AHR, you have a deficiency of ILC3s and also TA17. So you have a deficiency of lymphocytes that express Rho gamma T. And knowing that, that HR is important for inducing the cells, we then crossed these mice with Rho gamma T3. So we took fox mice for the transcriptor factor AHR and crossed them with Rho gamma T. So all the Rho gamma T expressing cells will deplete AHR, and eventually they won't survive because we saw the AHR is important for ILC3 generation. And we confirm this by flow that we don't have ILC3s anymore, as you can see by CD45 intermediate and type 1 high. Knowing that these mice don't have ILC3s, we infected them with C. diff to see what happened in the early infection. So as expected, these mice are more susceptible to the infection because we already know by our previous paper and by other group paper that ILC3s are important for the healing phase. And we saw that they don't recover at the end of the infection. You see here, mice are still sick at day 5. And they also have more bacterial burden on the tissue, suggesting that they are not able to clean the tissue through healing from the infection and recover. But we still don't know about the early phase. So what we did next is to investigate what happens in the infiltration of inflammatory cells in the lamina propria. So we investigate all the cells that infiltrate in this tissue after the infection. And we notice that among all the myeloid cells, neutrophils are very decreased in the ILC3 deficient mice, suggesting that locking ILC3s, we also lock somehow neutrophils infiltration in the tissue. And this phenotype is very specific to neutrophil, because if you go for dendritic cells, eosinophils, macrophage, we really didn't see any big, considerable change. The main change we found was the neutrophil reduction, less accumulation of neutrophils in the lamina propria. Then, very intrigued by this, less neutrophil, we decided to investigate if this neutrophil was, somehow impaired. And then checking the neutrophil maturation stage, we noticed that they express less CXCR2. There's a very important chemokine receptor related to the neutrophil migration to the tissue. And as you can see, ILC3 deficient mice express less CXCR2 in the neutrophil population. They also have less grams on the cytoplasm, suggesting impaired maturation. They also have less maturation by CD11B low expression. So when they express less CD11B, they are not ready for fighting, so they are immature. So we can see that we have more CD11B low neutrophils in the knockouts, mainly in the early phase. And also CD177, that is a marker for neutrophil activation. So we see that ILC3 deficient mice has less presence of this marker that is related to neutrophil activation. So overall, we see that we have less neutrophils and impaired maturation of these neutrophils in the ILC3 deficient mice. And it was very specific to ILC3s because we know that IHR also affects J17, but when we check neutrophils in the REG1 deficient mice and as well TCR beta deficient mice, they will lock T and B cells, and TCR beta will lock only T cells. We cannot see this difference in neutrophil. So having or not having T cells won't matter for the neutrophil accumulation, but not having ILC3s, then we have impaired neutrophil accumulation. So I was stressing that it was very specific to the ILC3 presence. Okay, to prove that the ILC3 was the really reason of this neutrophil impaired accumulation, we just restore the ILC3s in the ILC3 deficient mice. So we took the ILC3 deficient mice and we gave them ILC3s by IV. And then after 2 weeks of reconstitution, we infect these mice. So now the ILC3 deficient mice have ILC3 back. So infecting these mice, surprisingly, these mice was protected now. So they were protected, so they are not susceptible to the infection as the orange line here. So we can see the blue line. So having ILC3s back make the ILC3 deficient mice protected against infection. And you can see that this was really functional because we can see ILC3s back to the tissue. And most importantly, we can restore the neutrophils accumulation and maturation. So now we can really point out that ILC3 was important for protecting the mice and it was somehow related to the neutrophils. It's very well known in the literature that neutrophils are important for early C. diff infection. A lot of people have shown, and especially these 2 papers from the same group from Japan, that they show that depleting neutrophils you make the mouse more sick to C. diff. And it's because neutrophils are very important for sensing the C. diff toxins, and it creates like an environment that will contain any commensal bacteria that translocate to the bloodstream, avoiding systemic infections and avoiding death. So neutrophils are very important for the early infection. And now we know that ILC3s was inducing these neutrophils. So probably this is the reason why they are more protected when you have ILC3s and why they are not when you don't have ILC3s. But how? We still don't know. We know that there is a very strong correlation, but we still don't know how ILC3s induces this neutrophil response. For checking that, we did the obvious. We put these two cells together. So we sorted ILC3s from the gut, from the lamina propria, and we put in culture, co-culture, with neutrophils from the bone marrow that they are still immature, so they're still under maturation process. And then we wait for 4 hours to check what happened with these neutrophils. Similarly, to identify something that was secreted by these ILC3s, we stimulate this ILC3s from the gut in vitro, ex vivo, for 3 hours, and then we use the supernatant for treating the neutrophils from the bone marrow in this ex vivo manner. And then checking neutrophils in both conditions, we notice that having ILC3s in co-culture or only having the stimulation of these neutrophils by the supernatant from the ILC3s, in both conditions, we have more neutrophil maturation and activation, as you can see by CD11B and CD177 expression. So ILC3s was in fact secreting something that was activating this neutrophil maturation. Again, what? So we decided to check everything that was being produced by ILC3s. So we performed bulk RNA-seq on unaffected and infected ILC3s, and you check the main cytokines that are involved with neutrophil responses. Of course, all of cytokines are regulated, because if you have uninfected mice and you compare with infected mice, a lot of things will be up because inflammation will prime the cell activation. So it was not really telling much. We confirmed this information by checking flow, but as you can expect, all cytokines mainly were up because the cell were activated. So again, we need to pick one and trying to investigate what could be being released by ILC3s that is priming neutrophils. One cytokine that is produced by ILC3s is GM-CSF. And GM-CSF is very well known that induces myeloid production and maturation, and specifically neutrophils. So you can see that neutrophils are more primed to degranulate, produce ROS and phagocyte when you have higher GM-CSF levels. In a previous paper from Mir, they showed that ILC3s from the gut produce GM-CSF in steady state condition. That is very important for priming macrophage and dendritic cells. They are very important for the oral antigen tolerance. Knowing that, we decided to check, is GM-CSF being produced by ILC3s under C. diff infection? So we check at day 0 when mice are not infected, but they were treated with antibiotic. And then comparing with day 2 and day 4, you can see that there was a very high release of GM-CSF by ILC3s on day 2. And checking the whole tissue GM-CSF content, we see in the ILC3 deficient mice, we have pretty much less GM-CSF compared to the wild type. So ILC3 produces GM-CSF, and not having ILC3s, we have a deficiency in GM-CSF. And to see if GM-CSF is important, we infected GM-CSF knockout mice. And as you can clearly see, they're very sick on the early stage. So they are like susceptible to the infection right after the gavage. They have more bacteria, and they also have more bacterial translocation. So showing that not having the lack of GM-CSF impairs the early innate immune response to the infection. But is this whole body knockout mice related to GM-CSF produced by ILC3s? So to point out this, first we check in vitro. We confirm this crosstalk between neutrophils and ILC3s in vitro, cultivating ILC3s with the supernatant that from ILC3 stimulated with cytokines and toxin considered. And we treated these neutrophils from bone marrow with the supernatant in the presence or absence of neutralizing GM-CSF antibody. So neutralizing GM-CSF, we saw that when you have the supernatant, as we saw before, you have more neutrophil maturation. But then when you block GM-CSF, from the supernatant that was released by ILC3s, you cannot see this maturation anymore. So GM-CSF is important for inducing this ILC3 associated maturation of the neutrophils. And then the same we observe when we just co-culture white type ILC3s with neutrophils or GM-CSF knockout ILC3s with neutrophil. So having GM-CSF production by white type and not having by the knockout, we can see that ILC3s respond, neutrophils respond in a different way. So having GM-CSF production, you have maturation of the neutrophils and activation, and not having lacking GM-CSF, you don't have the same maturation as the neutrophil without stimulation. Okay, but this was in vitro. We really don't know if it was happening in the mouse as well. For doing that, we use the same adaptive transfer approach we used before, but now, instead of transferring white type ILC3s, we also have a group that the mice receive GM-CSF knockout ILC3s. So now basically we have a conditional knockout mice where you have ILC3 deficient mice that receive white type ILC3s producing GM-CSF, and another condition that we have GM-CSF knockout ILC3s, locking GM-CSF only in the ILC3 compartment. And surprisingly, when you have GM-CSF production by the green line, you see protection. But when you don't have the red, you see that these mice are very sick. They are not able to recapitulate the protection. So suggesting that expression of GM-CSF by ILC3s are very important for this early immune protection in the infected mice. And this was somehow associated with neutrophils. But if you check in the white type, having ILC3s, we have a pretty much higher amount of neutrophils. But then when you don't have GM-CSF, we don't have differences in the neutrophils. So GM-CSF was the reason why we are having more neutrophils infiltration in the lamina propria. The same was observed when we treat the mice with GM-CSF, recombinant GM-CSF, or neutralizing anti-GM-CSF. When we neutralize GM-CSF, they are very susceptible, they die. So you see all the mice die, they are very sick, and they have like a lot of, so you can see by the score, body weight, and survival. And when you treat these mice with GM-CSF, even the ILC3 deficient mice treated with GM-CSF, that is in red, they recover. So having GM-CSF, even in the absence of ILC3s, was related to CGI protection. And just to summarize what we have and kind of put everything that we notice in ILC3s in a physiological context, we know that we have three main subsets of ILC3s. We have one NKp46 positive, one that expresses CCR6, and another subset that don't express any of these receptors. It's called double negative. The GM-CSF is mainly known by being produced by NKp46 ILC3s. So we decided to check, was this subset the main source of GM-CSF, or was GM-CSF produced by another subset under infection condition? So when we check the GM-CSF producer, basically by gating whole ILC3s, GM-CSF positive cells, and checking the expression pattern of this receptor, we notice that the main source of GM-CSF was the NKp46 positive ILC3s. And we also notice that these cells are the main one accumulating in the early phase. So you can see when you have the infection, and the green line that represent NKp46 ILC3s, this goes up. And then only after 3-4 days, the other subsets goes up. So it's basically suggesting that double neg and CCR6 positive are important for the late phase, and NKp46 positive ILC3s are important for the early phase. And then comparing the cells in the GM-CSF level in the day 0 and day 2, you can mainly see the NKp46 positive ILC3s are the main one producing GM-CSF, and not the negative ones. For pointing out if it's really the case, we generate deficient mice for NKp46 ILC3s. So basically we cross ROR gamma T flox mice with NKp46 pre mice. So whatever express NKp46 will deplete ROR gamma T. So it will impact for sure the NKp46 positive ILC3s. So they will be depleted, they will die because they won't have the organomery transcriptor factor that is very important for the primary function of the cell. And we confirm by flow that these mice don't have ILC, this subset of ILC3s. And in fact, in these mice, we notice that they are also sick. They are also susceptible to the infection. And then we check all the parameters that we have as bacterial burden, GM-CSF expression, neutrophil maturation, and activation. We all see that this criteria were downregulated on the day 2 post-infection. But then this inflammation was unspecific and too much strong on day 5 post-infection, suggesting that NKp46 ILC3s are the main ILC. Of course, I'm not saying the main because we still have ILC1s, but among the ILC3s, NKp46 positive ILC3s are the main subset that respond to C. diff infection in the early phase and release GM-CSF that is very important for inducing maturation, migration, accumulation of neutrophils in the lamina propria. I didn't talk today about the production, but we also checked in the paper the bone marrow maturation and production by granulopoiesis. And we also saw that the ILC3 deficient mice have impaired granulopoiesis, and overall, this induction of neutrophil production and activation and accumulation is important for containing the infection and protecting the mice in the early stage. And then we have the late stage that ILC3 will release IL-22. They will be very important for the healing phase of the epithelial, damaged epithelium. So saying that, I want to acknowledge my lab for support me. Many of this data was possible through lab meetings, discussion, interaction with my lab mates. And I also want to say thank to Marco Vinolo from University of Campinas. He was my